All right, so today 8.4 transcription. Transcription converts a gene into a single-stranded RNA molecule. So we learned yesterday about genes, before yesterday too, but a gene is a 100 to 1,000 nucleotide long segment of DNA, which codes for a protein. A gene, in order to become a protein, has to go through a few steps. So the first step is transcription, where the gene is converted into RNA. And the RNA is then what converts into a protein. So transcription is the first step of a gene making a protein. And so you have DNA, double-stranded DNA, which is used to convert into a single-strand RNA, and that RNA is then used to make a protein. And that gene is then expressed that way. Did you get that down? And that's really, when we talk about gene expression, that's what we mean. Genes making proteins. That's how we get our traits. Various proteins give you the traits that you see. Okay, so let's go through the differences between RNA and DNA. You're going to want to, you don't need to draw what you see in here, but you do want to write down these right here, that RNA has these 4 bases. The DNA has these 4 bases right here. So you do want to understand how they're different from each other. So we talked yesterday about the 4 bases for DNA: cytosine, guanine, adenine, thymine. RNA has 3 of those 4, but instead of thymine, RNA has uracil. And that's the difference between RNA and DNA. RNA has uracil as the 4th base, and it acts like thymine. So for an RNA strand, whenever it sees A, it will link up with U. So U links up with A in RNA, but for DNA, A links up with T.
Holly. Miss Holly.
That's the biggest difference between RNA and DNA. The other difference is you have a single strand for RNA, double strand for DNA. So because of that, RNA is more easily broken down and mutated. DNA is more stable and less prone to mutations. And so that's why we use DNA to carry our genes instead of RNA. It's a more stable form. If we had RNA instead, then we'd have more mutations.
Does RNA have a double helix?
No, it doesn't. So it is curved, but it is not double helix. Double helix would be 2 intertwined.
Does it not pair with anything?
It can pair, but it won't stay as double helix. We'll talk about that in a second. So it does pair up in some instances, but not like DNA where it's always paired up. Oh, Christopher, this page, just write down the letters that RNA has these 4 nucleotides right here: C, G, A, and U. DNA has C, G, A, and T. And then also write down that RNA is a single strand, DNA is a double stranded. And that's all you need to know for that one. If I have this down. Okay, so I mentioned this, but we'll go through this in more detail now. So RNA carries DNA's instructions. There's a central dogma in biology which says information flows in one direction, from DNA to RNA to proteins. And that's what we see described right here in this picture. DNA in the nucleus, makes RNA. RNA moves out of the nucleus to the cytoplasm, and it makes a protein out there. So there's two major steps. Transcription is that first step going from DNA to RNA. Did you learn about dogma in theology class? Like what, church teaches dogma, and what that means? What did they say about what dogma is?
An official teaching.
An official teaching, right? Something that the church is sure about. And that's why biologists use this term. I mean, it's really a religious term. It's really not appropriate to use in science. But they used it because they were just wanting to say that we're just so certain about this. It's a dogma. And that's what they were trying to get across with using that term. But it's really not a good term to use because dogmas are for religion, not for science.
So DNA isn't converted into RNA and makes.
It makes it, well, I mean, yeah, that's right. So you still, at the end, after this happens, you still the DNA strand. It also has made RNA. So you now have two things. You have DNA still, and you have RNA that's made from the DNA.
I see.
So there's three parts to this. We talked about replication yesterday, where the DNA just copies itself. And then transcription, we're going to talk about today, where the DNA makes RNA. And then tomorrow we'll talk about translation. Actually, Friday we'll talk about translation, where you then use RNA to make protein. RNA is a link between DNA and proteins. So there's a middleman between DNA and proteins, and DNA has to convert into RNA first before proteins are produced. RNA differs from DNA in three ways, and I mentioned all these before, but we'll just go through them in more detail. The first way that DNA is different from RNA and vice versa, DNA has deoxyribose as a sugar. RNA just has ribose as a sugar. And so RNA stands for ribonucleic acid. Ribo is ribose sugar. DNA, deoxyribonucleic acid for the sugar that it has, deoxyribose.
Isn't that part of like the backbone? Or is that like it?
Exactly, yeah.
How it's stronger?
Exactly, yeah, sugar backbone. For both RNA and DNA, the sugar's in the backbone, and that makes it strong. Exactly. And as we mentioned, RNA has uracil instead of thymine as a nucleotide. Caleb? Okay. RNA is single-stranded, and DNA is double-stranded. As I mentioned too, RNA is not as stable as DNA. RNA breaks down more often, it's more prone to mutations. And that's why it's not as good to use in genes as DNA is. She'll have this down. And so as I mentioned too, don't write down this top one, you already have that down, but A goes to U in RNA, and then G goes to C, just like DNA. Transcription copies DNA to make a strand of RNA. Now, transcription, though, only copies one gene, not the entire strand. So in replication, you're copying the entire DNA, all 3 billion nucleotides. But for transcription, the idea is to just have one of the genes expressed. And so it's going to only copy one gene and use that, convert it to RNA, and use that to make a protein. We talked about DNA polymerase yesterday. DNA polymerase is what separates the DNA into two strands and makes it replicate. RNA polymerase is what causes transcription to happen, where you then convert DNA into RNA. So RNA polymerase is this tan blob right here, and that unwinds the DNA and then makes RNA. RNA polymerase, other proteins form a transcription complex, which this whole thing is right here. The nucleotides out here, and then RNA polymerase around the DNA, pulling it apart, bringing nucleotides that correspond, and making RNA.
So this is a different type of polymerase, and this is not during replication.
Yeah, that's right. So replication would be DNA polymerase, and that makes more DNA. RNA polymerase makes RNA. So RNA polymerase only goes through one gene. It just goes through, 100 to 1,000 nucleotides long and just stops with one gene. So this complex recognizes the start of a gene and unwinds only the gene in the DNA. It just goes through one segment of the DNA where the gene is, and it stops after that. Did everybody that one down? So nucleotides come in and they pair with one strand of DNA. So you see that this one strand of DNA is what is being paired up. This one's not being paired up with nucleotides. So it moves in one direction and it pairs up only on one side. And RNA polymerase is what binds those nucleotides together to make this red strand you see right here, which is the RNA segment. And then after it's done, the DNA just keeps on winding back up again after it passes through. Kind of like a zipper, part is unzipped and then you move it back up, it zips up again. And so it just brings in the nucleotides that correspond, so if you see a T on the DNA, it brings in an A. A C on the DNA, you bring in a G to make the RNA. But one difference we talked about before, if there is an A on the DNA, then it brings in a U for the RNA. Shall I write that one down? And then after the gene is transcribed, the RNA detaches from the gene and it goes out from the nucleus. And the RNA polymerase just breaks down. So the RNA now from that one gene has a message, which goes out to the ribosomes, which are factories to make protein, and it makes the proteins. There's three kinds of RNA that are made from transcription. The first kind is messenger RNA, also called mRNA. And that carries a message that will be translated to the form of protein. And so basically it comes from the gene and it gives a message to the ribosome to make the protein it calls for. Yeah, we'll see.
Aren't they using like messenger like RNA to make the vaccine or something?
Oh yeah, exactly. Yeah, that's right. Yeah, that's a good catch. Yeah, that's right. So the new vaccine for COVID is putting RNA into your body. And what does RNA do? We talked about that RNA makes proteins. So what it does is it's really interesting on how this works. It tells your body how to make a spike protein. And this spike protein is on the COVID virus. It's like the key that unlocks your cells. And so when the virus can get into your body and get into your cells, that's when it becomes infectious and hurts you. And so your body recognizes that key, that spike protein. And if it can target that and destroy it, then the COVID virus can't get into your body. It can't get into your cells. It doesn't have the key anymore. And so the vaccine has RNA instructions to make that spike protein. Your body sees that, it fights it, it destroys it, but it has to learn how to do that. And so it takes about a couple of weeks for it to learn how to destroy that spike protein. But once it does, it remembers that. So if you get the real COVID virus, the body can see the spike protein, it destroys it immediately, and you don't get sick from that virus. So they've never done this before. In the past, vaccines are usually dead viruses that your body learns how to attack. But this is the first time I ever used RNA to do this, where there's no virus, you don't get a virus. You just get RNA, which tells your body to make proteins. But so it sounds very safe to me because you're not getting a virus, you're only getting RNA. And as I mentioned, RNA is very unstable, it breaks down very easily. And so RNA will not be in your body for very long. It's going to just break down after a few weeks. So it won't stay in your body. So there should be no lasting effects of this. Now, if you have allergies, that could be one thing that could happen. Allergic reactions to the vaccine have been noted. But without allergic reactions, it is a very safe vaccine because you don't have genes change, you don't have viruses in your body. Like,
How do they know that it works if they like just made it?
they have tested it on a lot of different on animals, cells in laboratory, and they had human volunteers too. They had about 20,000, somewhere in the 10,000s volunteers before they have it on the market. So of course they can't be certain about everything, but they have had a lot of testing done, and all the testing has shown it to be pretty effective. As long as they can see that you make that spike protein, your body can attack it, then we know it's going to work because that's what you need to have happen. And that's what we have seen. Now, of course, is it perfect? Well, nothing's perfect, but it seems to work pretty well. Ribosomal RNA, also called rRNA, forms part of ribosomes where proteins are made. And so this is what makes up the ribosome, which is the protein making factory. And a transfer RNA called tRNA, this brings in amino acids, which are what make up proteins. These are in the cytoplasm. So when you eat anything with protein in it, you break down that protein into amino acids. They're floating around in your cells. ribosomes pull them in to make new proteins. Everybody have these down? Okay. Transcription and replication. These are both pretty similar. You know, you're copying DNA. It's just that for transcription, you're only looking at one gene and you're turning that DNA into RNA. But they both use a complex enzyme. Replication uses DNA polymerase. Transcription uses RNA polymerase. And they complementary base pair. That means that, you know, if they see an A base, they add a T, and that's for DNA. But if they want to make an RNA, when they see an A base, they add U. If they see a C base, they add G. So it's pretty cool to see this happen, transcription happen. You can see it looks like a feather where you have, this is one gene on your DNA strand, and you have a lot of RNA polymerases that are branching off and building RNA. And so all these little, it looks like little feathers on here. Each of these little lines is RNA being made. And so hundreds of them happen all at once. You have hundreds of these within one DNA happening all the time.
How do you know, how does the body know like which RNA sequences to go with? If there's so many of them?
Oh, like, you know, as far as like the ribosomes, because they're sent out of the body into the ribosome then knows what to do with it at that point. The ribosome then has, it's a machine, it's a factory. It can pull in the RNA and it has instructions and it tells the ribosome how to make the protein. So these are in the nucleus, these are all sent out of the nucleus into the ribosomes. The ribosomes then know what to do with the RNA at that point. Now the difference is this though, as I mentioned, replication only copies, well, it copies all the DNA and it makes DNA. Transcription only copies one gene. So while replication goes through the 3 billion nucleotides, transcription only goes through, you know, 100 to 1,000 or so nucleotides through one gene. Sorry. So write these ones down. And also replication only makes one copy. Transcription, as you can see, is making hundreds of copies all at the same time. Okay, that's the end of 8.4 homework today in 8.4. It's questions 1 through 5 on page 228.
Miss Holly,
Yes, go ahead.
can you send me the questions for?
Yes, I will do that.
Thank you.
That was number 6. All right, for those on Zoom, any other questions? I'll go ahead and send 8.4 to you through email. And then do questions 1 through 5 today for homework.
Thank you.
You're welcome. Have a good day.